Hi guys, Mr. Hyatt here. This is the Chapter 11 AP Biology lecture. Obviously by the title of the slide here, we're talking about cell communication. So first off, why do cells need to communicate? Number one, we need to regulate advanced cellular processes. We don't always need to be doing everything. We need to be able to turn processes on and turn processes off. So that's going to require some communication. We have to respond to external stimuli. Again, that's going to involve taking in stimuli. That's going to involve moving the signal to your brain, and then that's going to involve, doing something, responding. So then you see that third bullet point there that allows coordination of multi-step responses. Make sure you read about the reproduction of yeast. I don't have enough time to get in depth into it here, but I am going to talk about it just a little bit towards the end. But make sure that you understand or at least you've read that chunk before test day. So in terms of cell communication, a couple of different types of signaling. Notice here on the left side we're talking about local signaling. So one cell is signaling a neighboring cell. Then over here on the right side we've got long distance signaling. Maybe we need to send a signal from our hand to our head. So we're communicating over long distances. Notice that paracrine signaling is a strategy where we're going to make a whole bunch of whatever these ligands are, whatever these messengers are. We're going to make a whole bunch of them, and we're just going to excrete them. And the logic is we're just going to put the message out there, and our neighbors are going to hear it. Kind of like if you just blare your music really loud from your house, your neighbors are going to hear it. So if you're trying to get a message to your neighbors, that's a good strategy. But if you're trying to get a message from central Indiana to southern California, that's not a good strategy. So paracrine signaling doesn't work well for long distances. Short distances, it works pretty well. If you need to be more targeted with your recipient or who your recipient is, you're trying to get a private message to a neighbor. You don't want to broadcast that to everybody. You just want one neighbor to get that message. So synaptic signaling is going to be more like a telephone. You've got a direct communication from one cell to another neighboring cell. And we'll talk much more about that in Chapter 48. Same is true here of long distancing. We'll talk about that, especially hormones in our next subunit, Chapter 45. So cell signaling is a relatively new topic in bio, and especially here in AP Bio. It appears to answer a lot of questions in medicine, which that shouldn't be real surprising if these tasks take the coordination of multiple cells, multiple cell types and tissues and things like that, then it should be kind of logical that, hey, if we can figure out how to communicate, we might be able to change the codes they're sending back and forth. We might be able to change the end results. So there are 3 stages. Got to get these down. Reception, pretty easy. That's receiving the signal. Transduction, we're going to pass the signal along to somebody else. And then response is we're going to respond. That one should be pretty easy. Okay, so our reception typically happens at the cell membrane because that's where we're going to contact the outside world, the extracellular matrix. Excuse me. So we've got some sort of a signal molecule. Soon we'll call that a ligand. And we've got a receptor. We're receiving the signal by the ligand binding to the receptor. Basic summary of transduction, we're going to do something to pass that message along. We played telephone at one point in class. That's what we're talking about here with our signal transduction pathway. Now remember how messed up our signal got by the time we got to the end. So we must have a pretty accurate transduction pathway. And then last but not least, of course, response is going to be doing something in response to that stimulus. Again, reception is the target's detection of a signal coming from outside the cell, whether that's an external stimulus or whether that's a message coming from another cell. We don't know yet. That could occur through direct contact, through something like paracrine signaling, or that could happen through long distance like a hormone signal, something along those lines. Either way, still reception. Direct contact, I call this level B. I do think it's helpful to know what plasmodesmata and gap junctions are, even though I don't have a specific question on it on the test. Here's what we're looking at. Same exact thing, but you know that the structures are slightly different, so they get different names. Plasmodesmata is a hallway between two cells. Gap junctions, hallway between two cells. Just one's an animal cell, one's a plant cell. Could also be like receptor interaction. So here's like our macrophages in our immune system can recognize our host cells through receptor recognition. So this is how a white blood cell recognizes self versus invader. So again, that's just another type of direct contact cell signaling. Signal molecules. Notice that they're often water soluble and they're big, so they don't travel across the membrane. That's why we have to have a transduction pathway. That's going to be the actual chemical signal that travels from cell to cell. In class one day, I threw a ball from myself to one of the students. That was our signal molecule. I was sending that signal from myself to the student. They behave as ligands, which are smaller molecules that bind to bigger molecules. That's a vocab word. Hopefully you remember that one. Our receptors are usually made of protein, sometimes carbohydrates, but these receptors, in terms of cell signaling, tend to be proteins. They're going to change shape when it binds to a signal molecule. Again, form controls function. That shouldn't be a shock there. It's going to transmit information from the exterior to the interior of the cell without opening up a door, without exposing the internal contents of the cell to the environment. We've got four mechanisms. You really need to be familiar with each of these in terms of what they do. If you had to focus on two, I would focus on G protein and tyrosine kinase. Those are the really, really big ones moving forward. Now, I called this level B, even though I just said it's really important because, again, there's not a direct test question over it for me. So this is tested heavily on the AP exam, so definitely don't just breeze through it because I called it level B. Again, it's a plasma membrane receptor, the G protein. It works with G protein and an intracellular protein with GDP or GTP. Here's a picture. You know I talk better from pictures. So here's what we're talking about. Here's our G protein. Okay, so we've got this signal binding site. That's our true receptor where the lock and the key has to fit. We've got an acetyl group over here, and we've got an amine group over here. But GDP and GTP is basically going to act as like a light switch. So if we've got GDP attached to the G protein, then the switch is off. If we've got GTP, if we phosphorylate that molecule, now we've turned it on. And that should sound familiar, and it should definitely carry through the rest of this lecture. Phosphorylate something, you turn it on. So when it's turned on, it's going to bind to another protein, which is an enzyme. It's going to alter its activation. It's going to lower the activation energy. Remember all that stuff from our enzyme discussion. And it's going to activate the protein. It's going to fire and then close. We don't receive the signal forever. Okay, so here's our G protein with our GDP attached right there. We've got our G protein linked receptor, so that's the curly Q thing from our last picture. Here we're inactive. Okay, so then we phosphorylate our GTP. I got ahead of myself. Signal molecule binds, G protein moves over. As that happens, GDP is going to be displaced by GTP. That's going to turn this molecule on, and then whatever's going to happen happens. At some point, the signal molecule dissociates. The protein is going to travel back down the membrane. Very widespread and diverse in function. G proteins do a ton in our bodies. So that's kind of why I said it was so important moving forward, even though I'm not directly testing you on them. And it appears that a lot of diseases work by affecting the receptors to G proteins. You see some examples there. Up to 60% of all medicines do what they do by manipulating G protein linked receptors. Think about that. 60% of all medicines. You guys are all smart people, and you probably had no idea. 60%, more than half of our medicines are manipulating G proteins. Moving on to tyrosine kinase receptors, definitely an A-level thing. Extends through the cell membrane. The intracellular part functions as a kinase, which you should remember basically dephosphorylates ATP and turns on something else. In this case, it's a substrate protein we're attaching to a tyrosine. So the mechanism of a tyrosine kinase, the ligand's going to bind. It's going to cause our two receptor molecules to aggregate, which means come together. Growth factors do this. It's going to activate the tyrosine kinase parts inside the cell, and it's going to turn on those tyrosines. It's going to turn on those proteins. It's going to make something happen. So again, here's our picture. We get our signal molecules that bind. Our tyrosine kinase monomers come together and form, notice here, phosphorylated dimer. It's phosphorylated because we've added these phosphates. It's a dimer because we're putting two together. Di always means two. So monomer means one, dimer means two. So we've activated these relay proteins. Now our cellular response is going to happen. Remember, we're focusing on reception, so we're simplifying the transduction piece here. Those intracellular proteins are going to become activated and cause a cellular response. Remember that intracellular means they go through the membrane. Again, maybe not again, but these things often do regulate many pathways. If you see here, we've got multiple phosphates that we can, multiple tyrosines that we can phosphorylate and turn on multiple pathways all at once. Some of the less important mechanisms, ion channel receptors are going to be protein pores in the membrane. They're going to open and close. Think about in neuronal communication. We've got those neurotransmitters that have to be released. We've got things that have to be taken in on our target cell. So that's kind of what we're talking about. These ion channels are basically just going to open and close. They're activated by a ligand. It's going to cause a change in concentration inside the cell. Notice our nervous system cells signals operate this way. But again, we're just going to get a ligand that binds. It's going to open a hallway, and now ions can diffuse through. Once that cellular response happens, or these concentration equalizes, however you want to say that, the ligand dissociates and moves on. We will leave off here for lecture 1. We'll pick up here on part 2.